good morning, good afternoon, everybody, and welcome in from across the country and beyond for another exciting Exploring by the Seat of Your Pants broadcast. So my name is Jesse. I'm the host here, and as you saw from that intro, we're all about bringing conservation, adventure, and science into classrooms around the world. And one of our big partners over the last 5 years has been the amazing folks at the Gairdner Foundation. They work to showcase and celebrate the most amazing biomedical and global health researchers on planet Earth. Every one of our broadcasts with them are on our YouTube channel if you want to check out some amazing stories from some really stellar laureates. And I really can't encourage you to check out their website enough to learn about all the amazing work they do. This is the first of two amazing Canada Gairdner Awards virtual roundtables. We are bringing together some of the best and brightest in a wide array of fields to highlight and showcase their incredible work to make the world a better place. And so I'm so thrilled to get to be your host. Thank you so much for tuning in today from across the continent. And please do share your questions in the chat. We'd absolutely love to hear from you there as well. Now, without further ado, I'm going to bring in all our speakers just to say a quick hello and a wave, and then we are going to turn it over one by one to all of them before we dive in with a broader roundtable session of the broadcast. So, Dr. Greenwald, Dr. Stinson, Dr. Briand, Dr. Stroul, and Dr. De Carvalho, welcome in, everybody. Thank you all so much for being good sports and joining us for this virtual roundtable. You all look very scholarly today. We've got some great backdrops, which is kind of fun for me. I'm an aficionado of backdrops at this point. And I'll leave you in the background as a bit of a spoiler for now. And we are going to begin with videos for each one produced by our friends at Research to Reality. They do some amazing work too in showcasing cool scientists, so I encourage you to check them out. So we'll begin with videos one by one with each of them, and then we'll ask each of our amazing laureates a question. So without further ado, here's our first video for Dr. Jennifer Stinson.
My name's Dr. Jennifer Stinson. I'm a nurse clinician scientist at the Hospital for Sick Children. I'm a nurse practitioner in the chronic pain program, and I'm also a professor in the Lawrence S. Bloomberg Faculty of Nursing and the Institute of Health Policy Management and Evaluation at the University of Toronto. What my research and clinical areas are that I'm being recognized for is my work with young people with chronic pain. Chronic pain affects 1 in 5 children and adolescents in Canada. And it's often undertreated because healthcare professionals lack the training and education in pain. 70% of the time I do research, and 20 to 30% of the time I'm spent caring for children in the pain clinic with chronic pain. I love my job. I'm so passionate about it because every day I come to work, I see patients and families. I can address the questions that are so important to them through my research. So what I do in my lab is try to use digital therapeutics and virtual care solutions to improve access to care for young people that have painful chronic conditions. Because I'm embedded in a hospital and I work clinically with these patients, I can get my technological interventions embedded back into the clinic, so a generation of kids are not losing out on research that's been done.
Well, first of all, I think we can all agree we all want that robot in our emergency room from now on. So, Dr. Stinson, thank you so much for that great dive into a little bit of the work that you do. I recently had a broken ankle. I took up our national pastime as a 33-year-old man and rolled over it, and had to go to the hospital for the first time in many years. And we've got a swanky new hospital here in Corner Brook, Newfoundland, with all sorts of bells and whistles. And the thing that struck me most was that we could use technology to help book those things. I was able to talk to a doctor in advance through digital technology in a way that simply wasn't possible for my mom and my grandmother's generation before me. So I'm really curious, whether it's robotics or other things, how do you see digital technology sort of changing the patient experience in the decades to come?
Yeah, that's a great question, Jesse, and hope you're okay. I could give you some advice for pain management if you needed it. But since the beginning of my career, there really have been monumental changes in the accessibility and complexity of digital health technologies that are available. So, for example, when I first was doing my PhD, I created one of the first diaries to help young people with arthritis track their pain. And now I was using archaic technology, the PDA, those things don't exist anymore. But I think one of the most exciting things about my work is that I need to stay on top of cutting edge technology, because the young people that are in this audience are super savvy about technology. And so we need to make sure that it's really engaging. So I really see opportunities moving forward to really help improve precision medicine. So that's being able to get the right treatment to the right person at the right time. And I think some of those things will be using artificial intelligence and wearables. For example, you saw in the video that we were using a robot called Medi, and that robot actually is an AI enhanced robot. So it can actually hear and see what's going on in that emergency room. So if the child becomes really upset, it can pivot, right? Instead of trying to distract it from that painful procedure. So I think there's just going to be more opportunities to use AI to help personalize medicine. But I do know it's hard to predict what things will look like in the future for sure. But I do know that one of the most important things to me is that we really involve patients and families' voices in every step of developing technology, and also how it's implemented and scaled up to ensure that the value of that technology is really enhanced, and that we're making sure we're aware of the safety and privacy issues that young people are concerned about today.
Professional detachment out the window. That was a beautiful answer. Thank you so much for that. And I love that you highlighted the sort of the role of a researcher as someone who's had to be constantly on the cutting edge and constantly learning. I think when we're students, we think, oh, you finish grade 12, you're done, or maybe you're going to go to university beyond that. But the people that really seem to love their jobs the most, and this is true of not just Gairdner Laureates, but everyone we feature in this broadcast, just love learning and love being curious for their entire career. So thank you so much for that, Jennifer. That was wonderful. Oh, all right. We're going to be back to Dr. Stinson in just a little bit, but we are going to dive in with our second video. Again, we've got a lot of laureates for you today. It's a very exciting broadcast. So I'll cue up our second clip and we'll dive in with a question for them right after that.
My name is Iva Greenwald. I'm the DaCosta Professor of Biology at Columbia University. I work on notch signaling and cell-cell interactions in C. elegans, a microscopic roundworm. So in my work, I've characterized lots of mutations that affect that process, defining notch genes, notch signaling pathway components, and modulators of signaling. And the general significance of that is that because things are conserved in all animals, what we learn in something simple like C. elegans can be applicable to even human development, and mutations in notch and signaling components can explain certain congenital diseases. But also, aberrant notch signaling is actually a cause of certain cancers.
My name is Gary Strohl, and I am the Herbert and Florence Irving Professor of Genetics and Development at the Vagelos College of Physicians and Surgeons at Columbia University, and also a member of the Zuckerman Institute. My contribution to this body of work has been to understand what is spatial information, how is it generated, how is it received and interpreted, and how does it change cell behaviors to create patterns. My work on notch really relates to that last aspect of the problem, which is local communication for cells to coordinate their behavior. So the prize is for basically the discovery of that system, its understanding what its role is, and then ultimately getting to the molecular mechanism by which that system allows cells to communicate. And more than just communicate, cells have to talk to each other, but they also have to listen and change their behavior and talk back so that they coordinate, so they do the right thing relative to each other. For me, it's been very valuable to be paired with Iva, because we both have this deep conviction that this is an important way to understand life.
Super, super cool. Geez, I first remember hearing about notch signaling and how you can sort of do things with genetics in this book, which for our high school audience, I can't recommend enough, by Siddhartha Mukherjee. Just an amazing, amazing read. But I'm going to bring in both of you now to ask our question together. Welcome in, Dr. Strohl and Dr. Greenwald. So nice to have you both here today. When I went to university, there was this whole lexicon of Latin terms that I was unfamiliar with when I came out of high school. I heard about Drosophila and Arabidopsis and C. elegans. And so these are some of our sort of iconic model organisms that a lot of genetic and a lot of science research is done generally. So I'm really curious, what do you think makes them such a powerful system for discovery? And is there like a greatest model organism? Can you explain a little bit about what this means for our audience that might not know?
I guess I'll go first. So before I talk about my personal favorite, I want to say that I don't think there is really a GOAT, because you choose the model best suited to what you want to study. Mendel's peas, E. coli and its viruses, baker's yeast, worms, flies, and mice, all have had their moments of greatness, but what they all have in common is genetics, and that's no accident. So now for my favorite organism, the microscopic nematode worm C. elegans. I think it all goes back to the brilliant pioneer of molecular genetics, Sidney Brenner. He'd worked out fundamental features of the genetic code using bacterial viruses, and then he wanted to study problems in animal development and neurobiology using similar approaches. So he considered lots of options, including Gary's favorite, Drosophila, and decided to develop C. elegans as a model because it had practical features that he could foresee would enable fabulous genetics. And so, for example, if you put a single hermaphrodite on a petri dish, regular petri dish, with food and come back a week later, you'll see more than 100,000 grandchildren of that single worm on that dish. And so also it hit the sweet spot in between simplicity and complexity in its anatomy and development. So an adult hermaphrodite is a complete little animal with nerves and muscle, no bones. And it has only 959 somatic cells. And that number is precise because the cell lineage is essentially invariant. And because the worm is transparent, wonderful scientist John Sulston was able to sit down at a microscope, watch the zygote divide, their cells divide, etc., until the complete organism was formed. It's an incredible feat in itself, and it provides a clear baseline for applying genetics to understanding how cells know what to do. So those are the two features that first attracted me to C. elegans as a student and have been foundational for all the work I've done on cell-cell interactions, notch signaling, and cell fate.
What a beautiful answer. Gary, I hear you have a different one from that. We were spoiled that you have a different favorite, and sell us on it.
Okay, well, first I agree with everything Iva said about there not being a goat. Really, you choose a system because it lets you study the problem you're most interested in the most effective way. And also, you choose a system because the type of experiments you like to do fit with that system. In my case, there are two main reasons why Drosophila fruit flies are my favorite organism for discovery. First, I have a lifelong fascination with using genetics to understand basic biological phenomena. And what fascinates me about genetics is that it allows me to, in effect, reach inside the organism from the outside, to get right inside individual cells and change their working parts, the protein products of individual genes, and then look at what happens to those cells and to their neighbors. And over 100 years, after over 100 years of research in flies, we now have an incredibly powerful repertoire of tools for doing such experiments. And in fact, that repertoire gets better and better every year. And these tools allow us to identify most of the working parts underlying any given biological process you might be interested in, and to delve into both the logic and even the molecular mechanisms by which they work. And the special value of using such genetic approaches is that they tell you what's really happening in vivo, in the living tissues that will construct the animal. So rather than taking the animal and its cells apart to learn how they work, we learn by setting genetic traps that tell us what happens in living cells and tissues in the intact animal. The second reason is that the fundamental question I'm most interested in is the control of growth and patterning. And the incredibly powerful genetic tools we have in Drosophila are exactly what we need to discover how cells communicate with each other to decide what to do. My work with Iva together have in nematodes is an example of the complementary powers of these two systems to discover basic mechanisms that govern animal development.
Oh, So I don't think this is too much of a spoiler to say that going into this broadcast, this is not your usual thing, is talking to a bunch of students. And both of you were just dripping with enthusiasm about those animals. I'm a big animal guy. I like big toothy things in the jungle and in the big rivers in Australia. And so to hear such a passion about a worm and a fruit fly is just really, really special. And they are amazing creatures. For any student that's interested in genetics, you're going to hear these names endlessly in your entire education, starting at about grade 11 onwards. So thank you both so much for that beautiful answer. Oh, this is you three so far, scientists, have got us off to a fantastic start. We're only like halfway through. So I'm going to queue up our third video and you can learn from Dr. De Carvalho about his work.
My name is Daniel De Carvalho. I'm a senior scientist at Princess Margaret Cancer Center and professor at the University of Toronto. My work is work on DNA methylation or cancer epigenetics. It's based on the last 2.5 decades that we have been understanding DNA methylation changes in cancer. And the Gairdner Award is for our discoveries on how DNA methylation changes in cancer can induce transposable elements. Changes in DNA methylation can activate transposable elements such as endogenous retrovirus and other selfish DNA elements. Once those are activated, they can be recognized by the immune system as a viral infection. So we call this viral mimicry. So it's the concept that we can activate transposable elements in cancer cells, make them look like a virus-infected cell, and take advantage of these millions of years of evolution to identify viral infection and use this as an anti-tumor immunity. In addition to that, we have been identifying that this global change in methylation, which happens across all cancer patients, across all cancer types, provide a very rich source of biomarkers. They look different if they come from a normal cell versus if they come from a cancer cell. And then using these, we can use data science to identify if it's a cancer cell, a lung cancer versus a breast cancer. And if a breast cancer, even subgroups within the breast cancer. So this opens up possibilities of using methylation or cancer epigenetics for early cancer detection. This
So we had to edit the video. It was not like we missed anything there, just so everybody knows. I want to stress this realm that we're in now, where we're starting to learn how to hijack the immune system to do the things that we want against the things that we don't want in our bodies, including cancer cells, is just absolutely mind-blowing. Like, it's such a golden age of science exploration. So Daniel, thank you so much for that little overview of your work. And I'm so curious for you, starting a company alongside your research is a huge challenge. So we want to know what you've learned from the entrepreneurial side of science, because this looks like it has some really cool applications going forward. Oh, Daniel, your microphone's off. We've had our first tech snafu in the whole program. This is bound to happen. Someone has to have something go wrong with their tech. Otherwise, it's no fun. See, it was working just before we went live. So I find it's emotionally satisfying to smack the computer, even if it doesn't technically work. Your mic is on. What I'll suggest is this. If you want to leave and come back, we can go on to Dr. Briand, and then hopefully everything's kicked into high gear for our audio again, because you were working, as I said, mere minutes ago. So try that. Good luck, and we'll play another video and come back to that question in just a minute, okay? Thank you so much, everybody. See, something has to go wrong. We all lived through COVID. We've seen this ages ago. All right, while you're waiting for that question, while Daniel's prepping his entrepreneurial answer, we are going to play our program for Dr. Briand. Let's do that, and then we'll bring him in first question.
I'm Dr. Briand. I'm a medical doctor, and I've been working doing research on child malnutrition for many, many years. When I was in Bangladesh, I did research on the association between malnutrition and mortality. There must be something like 17 or 19 million of children who suffer from acute malnutrition. The real number, we don't know exactly how many there are, big number. At that time, WHO recommended to treat the children within the hospital with a liquid-based diet, which limited the capacity for treatment. Treating a child in hospital is always very expensive because you need a facility. It's expensive for the health system, but as well it's very expensive for the family. My contribution was, first I found that children who are going to die from malnutrition can be easily identified by measuring their mid-upper arm circumference with a simple tape. And also to develop, in collaboration with a colleague from the food industry, Michel Lescan, to develop a diet which had a similar nutritional property, but which could be consumed without the addition of water. The dangerous part of the traditional diets was the mixing of food and water, because food And water provides an excellent growth media for bacteria. If you eat them separately, if you eat the food without the addition of water and the water is taken later, you considerably decrease the risk of accidental contamination, and then this part of the treatment can be safely carried out at the household level, at the community. And this led to the possibility of massively increasing the number of children which could be treated.
How amazing is that? I want to say, before we dive into the truly like world-changing aspects of that, in these videos, we've had a lot of people walking around in Canada and the United States. That French street is like a great place, looks like a glorious place to have a stroll. Dr. Rion, thank you so much, and so nice to have you here today. So this ready-to-use therapeutic food, how widespread is it? How many lives have we saved with it? Like, it seems from people that are constantly watching people in dire straits around the world facing starvation, it seems like a really spectacular tool for helping millions and millions of people. Is there a sense we have of how widespread this is right now?
So we have an idea of how many children are treated every year, thanks to the quantity of which is produced and distributed. And we can say that roughly it's about 7-8 million children which are treated every year. The second part of your question is more difficult to answer. How many deaths do we prevent with this treatment? The honest answer, I think, is that really we don't know. My best answer would be quite a few, a lot. If you look at the UNICEF reports, UNICEF tried to have an estimate of the death averted because it's very important for them as well when discussing with donors. They produced recently a report where they estimated this treatment, community-based treatment of severe malnutrition, saves between 900,000 and 1.5 million deaths every year. I have limited confidence in this figure because the proper method to estimate the number of deaths averted is to have a group of children treated and a group of children untreated, and this will not be possible for ethical reasons. So I stick to my usual answer. Honestly, I don't know, but I think it's a lot.
It's really quite incredible. I'm just kudos to you. I mean, I know we're dealing with a lot of innovations today, but the fact that we're getting this to people in such en masse and saving lives in as effective a way as we can is really, really special. So kudos to you and thank you for this. I'm very excited for your answer to some of our joint questions in a minute, but thank you for our answer. Much appreciated. Daniel, we're back. We're going to try the tech. Fingers crossed, buddy. Here we go.
I hope it's working.
It works. It works. Christmas come early. All right. So tell us a little bit about the entrepreneurial side of science. Much appreciated.
All right. Thank you, Jess. Thank you for the introduction and sorry for the technical problem. I think from my side, I was focused on the scientific discovery. All my academic work was on the scientific and academic side. It's really about the curiosity, about asking questions, trying to understand how biology works, about creativity, how you approach this question, how develop experiments to prove or disprove the hypothesis. So that's have been always the side that I focus on my academic lab. Now starting a company was looking for the other side, is bringing the whole arc of development, taking this scientific discovery and now trying to develop a product that can help thousands, millions of people in this fight against cancer. So within the development side and the company creation, that's teach you a lot about other aspects beyond the creativity, beyond the curiosity. You start to think about the reliability of the product. Can this be generalizable? We're working in Canada. Does that work in the US, in Europe, in Africa, in Asia, with different genetic backgrounds? We start to think more about how robust this product is, how this product can be used to help people. So you think a lot about the safety side, you have to think a lot about the implementation side, not on the discovery side. So it's very interesting, the aspect that you work with these large multidisciplinary teams of scientists, biologists, but also clinicians, also engineers, software engineers, data science engineers. So you start to see this whole arc of different aspects that we need to translate that initial scientific discovery into something that can actually start to have an impact in the real world. So that has been very exciting for me to work on that side as well, be able to see from the early ideas all the way to discover and then all the way to create a product that have an impact on people's life.
What a beautiful answer. Daniel, again, I just, the human-centeredness of your work and of everyone's work in this virtual roundtable is really, really special. And the fact, there's a reason that Gairdner lauded you, all of you, because this work can, as you said, impact thousands, impact millions of people from stuff that starts in a lab. And I think that's one of the most exciting things about science, is that you can start off with a problem to solve or an interest that you have, and it can lead to truly world-changing results. Anyway. Thank you so much. What a beautiful answer. And we are going to bring in everybody in a second. I'm going to put you in the background, Daniel, for a second, but I'm bringing you right back. Before we dive in with our questions for everybody, and there's a bunch of really special ones that have been submitted by our friends at Visions of Science, one of the best networks for learning in the whole world. We're going to bring everyone in for joint questions. Please do share some in the chat as well. One of our notes in the chat for you, Iva and Gary, is fruit flies feel like such an enigma to me. It's incredible what you're able to find from such a small creature. So I'm glad, Jessica, in the chat, and anyone else who might be tuning in from our Ottawa virtual school and beyond, if you're taking something from these, this is what it's all about. This is why we have these virtual roundtables. So thank you for being such a great audience. All right, I'm going to bring you all in. It's going to be crazy. We've been one by one. It's been so polished. Now we're going to make it crazy. 6 people live on camera at once. And we'll begin with my favorite question in all of science and the hardest question in all of science, which is, what's the big idea? What, for students that are starting out today, what most excited about? What should they be diving in on? What's a problem that you think would be most exciting to solve? So because you happen to be at the top, Gary, you get to be first to answer this, but I'm going to leave you all on camera to nod along with one another as we do this. The big idea.
Okay. Well, for me, I think one of the most exciting questions are what I like to think of as the hidden mysteries that are in plain sight. And for me, the question that's most interesting, and which I think will be interesting long into the future, is just look at your 2 hands. They're the same size. Even though they're composed of literally billions of cells, they have the same size, they're the same shape, you have the same number of fingers on each hand, very similar patterns of hairs and nails and veins and muscles. So the question is, something has to be governing the growth and the patterning of these 2 organs so that they are virtually identical. So that's a fundamental question. And I should say we've learned a lot by looking at simple systems like bugs and flies, where we've discovered many of the signaling systems that allow cells to communicate with each other to tell them what to do and where they are. And notch signaling, which we received the award, is one of those systems. And we also now know that those same systems work in basically all animals, including all of you. So the problem is we still don't fully understand how they control patterning. And frankly, in terms of growth, we have no idea, even to this day, how they control your organization, how they organize growth. So looking at your hands, why did they stop growing when they reach the right size? This is a fundamental and remains unanswered. So I've been fascinated by these questions for the entirety of my scientific career. And I think these questions will continue to engage and fascinate scientists, maybe even some of you, for many years to come.
Very cool. Thanks, Gary. Strong start. Daniel, you're on our same line here, buddy, so you're next.
Well, I have 2 advices. One for the students to think about their own questions, find what they find interesting, even if it looks like very niche and only you are interested in this, you can make new discoveries on that specific niche and become something much bigger. So don't pay attention only for what we are saying here. Think about your own question and your own niche interest. On my point of view, I think we are entering this era of predictive biology, predictive health. So the big question for me is how we use all these molecular signals that we can use, all these physical signals like wearables, molecular diagnostic, all this information that we can get for every single person, every day, every time, and start to create models that can predict health as moving forward. So can we start to predict in a personalized manner when someone is going to get sick before they feel sick, what type of disease they might get in a month from now, in a year from now, 10 years from now, and how can you use this to intercept? How can you use this to prevent disease? how can we interact with the disease much before the problems happen with that. And this type of question, we need to have a very broad mindset. It's not only biology, it's not only computer science, it's not only engineer. You have to think this in a multidisciplinary way. And I think especially for the young audience, that's perfect for them to have the opportunity to build in multidisciplinary fields, not being fenced in a single discipline, and actually think more broadly than that. So that would be the biggest question in my mind.
Very, very cool. And I love the interdisciplinary approach. I find, through the history of science and technology, whenever we bring together people from a wide array of disciplines and put them together where they can ask questions of one another and sort of make bigger discoveries jointly than they could separately, we have the biggest leaps in science in the world, whether that's corporate, whether that's university. Countries have done this at certain times in history. So I love that message for our kids that might not know quite what they're particularly keen on just yet. Thanks, Daniel. Again, I'm just blown away by your answers. It's like you guys are all trained science communicators, which is really hard to be this polished in answers. So way to go for all of you. Andre, if you want to come on in, you're good to go. What's the big idea?
I think, all my work, all my life, I work to prevent death from associated with malnutrition. But I think we should go beyond that for the next generation. There's an urgent need to try to prevent the children becoming malnourished. And there are several possibilities to improve current situation. And also for those who become malnourished, we should go beyond survival. We should look at the quality of their life. These children later on in life, they have several problems. Very often when they become adults, they have a higher risk of getting chronic disease, including obesity, diabetes, and so on, hypertension. We should better understand the mechanisms behind that and to find ways to prevent this unfavorable outcome. And although some of the children, they have problems when they go to school, they have some delay and so on. And we should try to, again, not only to have them survive, but to have them thriving and becoming bright as other children.
It's such a nice answer. It's one that for our Canadian and American audience, we're not really used to thinking about childhood as being a time where you could be starving. I mean, certainly I grew up in sort of middle-class Toronto. Everything was available to me. Oh, we go to the grocery store, we get whatever we need. We never had to worry about that. But for billions of children, that's not the case. And so things like this, where we can make sure that people are healthy from the get-go, are really, really important. Weird parallel as well. I come from the world of conservation, and the focus for years has been, oh no, we're at the last two of an animal. We better try and save it now. And the focus now has been shifting to, hey, let's have a future of abundance. When there's a million of something or a billion of something, that's when we need to start working to protect them. And the same applies in a wide array of scientific disciplines. So thank you so much for that answer, Andre. Much appreciated. Jennifer, come on in. It's been a while since we've had you on.
Thanks, Jesse. So I'm going to build off Daniel. I think I'm really optimistic that we're moving towards a future where technology will enable us to provide more precise and personalized healthcare. But I think there's a big but. I think a critical question facing the next generation of young researchers is, how do we ensure that the application of the technologies that we're developing or using remain compassionate and human-centered? And perhaps even more important from my point of view is, what are the methodologies and policies that we need to have in place to make sure that when we develop these technologies, that they support equitable improvements in health outcomes and quality of life for all patients, especially equity deserving populations, I think is really critical.
That's a really, really thoughtful answer. My wife's a librarian at a university, and so every class is calling her in to be the AI expert and talk about the future of this technology. That's all she does now. And it's been really interesting seeing students start to go from, hey, this is a universal good, to, hey, there are some problems inherent in some of these technologies that we need to address now before they become entrenched. And so a sort of golden good example of AI and technology in medicine is Google DeepMind and AlphaFold, which won the Nobel Prize a couple of years back or last year. So I encourage our classes to check that out, because that's where it's been done really, really well and is going to lead to some big pharmaceutical and other developments over the decades to come. But human-centric, Jennifer, is our guiding principle. Thank you very, very much for that line. I have a last but not least, I promise, and then we're going to take some student questions. You guys have been fantastic. Come on in and take us away.
Well, I'd like to build on something else, excuse me, that Daniel said, sorry, which is that really there may not be a single most exciting question. That maybe the most exciting question is the one that inspires you to understand something. And many important discoveries have been made by scientists who just began by being curious about some natural phenomenon and then did careful and rigorous studies of it, and it led to things they couldn't even imagine. And so I just want to give an example of a simple question that led to amazing and unexpected things. I think some of you may have heard about restriction enzymes and CRISPR.
Yes.
Restriction enzymes are molecular scissors that cut DNA and are used for things like gene cloning and genetic engineering. And CRISPR is this amazing mechanism for targeting changes in the genome. So you could change genes inside of people, in fact. And there are now CRISPR-based treatments for things like sickle cell anemia and clinical trials for scourges like cancer and infectious disease therapies. But these amazing tools, restriction enzymes and CRISPR, weren't discovered because people were looking for those kind of tools. They were discovered when people asked a simple question: how does some bacteria resist infections with viruses? And because I believe past is prologue, I think we can expect some great science for the young people listening who begin their journey by following up just on something they were curious about.
You used my golden example. So we get questions from students all the time in our broadcasts, like, why do science that sort of doesn't have an end game? And this is something that governments have dealt with for decades. I mean, there's governments right now that say, hey, if there's not a concrete, tangible outcome to this, why would we do it? CRISPR is one of the golden examples. You can look at addressing the atmosphere of Venus, leading to CFC banning around the world and saving millions and millions of lives. Like, fundamental, pure research, knowledge for its own sake, following those questions that are of interest. If you take away nothing else from today's broadcast, I guarantee you our 5 laureates here today and pretty much every other scientist we're ever going to feature start with that as their guiding principle. So that is a beautiful answer. I've much appreciated. I also want to note for everyone too, if you're really keen on science of all kinds and you want to follow that principle, another great book recommendation, A Short History of Nearly Everything, my favorite science book of all time. If you 5 haven't read it, you should read it too, because it's really, really great. All right, we're going to dive in with some amazing student questions shared from our friends at Visions of Science Network for Learning. They're based in Toronto. They are my favorite organization in the world at doing this. So I'm so glad they had the chance to film some great some of their students. So we're going to play these. And because we've got about 7-8 minutes left, what I'll do is I'll pick a few of you each time to answer each one of the questions. But let's pull this up and get underway.
Could give advice.
There we go. Why did it freeze? There we go. In theory,
To your younger self as a student, what would it be? What first inspired you to become
a scientist. I don't know why it doesn't like to play nice with me right now, but if you could give advice to your earlier self and what inspired you to become a scientist, what would that be? So I'm going to start with you. Actually, we're going to go in reverse order. You and Jennifer can field this one for us.
Okay. Well, in terms of what inspired me to become a scientist, I grew up in the 60s, and the big thing was the space race. And I had fantasies of being the first child astronaut because I had this book called How and Why Wonder Book of Planets and Interplanetary Travel, and I memorized it, and I thought NASA would be so impressed by how much I knew that I could go to the moon. And then when that didn't happen, I got another book called The Human Body, which is where I first learned that we came from a single cell and look at us. And I got interested and read a lot of science books. Another influential book was The Microbe Hunters. So, I can meld the Human Body book and the Microbe Hunters book as being really foundational for the kind of stuff I'm doing now. And I think there was another part to that question.
Yeah. What advice would you give to your younger self? Like if you were coming up in science, what's the bit that you'd tell them to do?
Yeah, I think just get the best training in basic science you can get, and then you'll be prepared for anything, whether it's continuing doing basic research or something more applied. I said applied.
Very, very nice.
Maybe working on a fly. I don't know. And I think
whenever we can.
Yeah, and one of my former students has been extremely successful in developing drugs.
Yeah,
including the first drug based on a phenomenon called RNAi RNA-mediated interference. And he's told me that he feels that his basic science training has been the key to his success. So I think it opens up any door.
I mean, if he got his basic science training from you, I have no doubt. Okay, it's very, very nice. Thank you for incorporating some books into that answer too. As you can tell, I'm very literary minded, so I appreciate it. Jennifer, what's your answer?
Sure. I think what got me excited about science was when, as a new nurse, I really saw that in the hospital we were really under assessing and under managing children's pain. And I had this moral distress because what I was learning in school was not what was happening in practice. And that's that classic knowledge to practice gap. And so that really sparked my passion for going back to school and learning how to conduct important research. And I think the advice I'd probably give my younger self would be really prioritize building relationships. I think a lot of us have talked about, science is like a partnership with different disciplines to really address the most important questions. And I think for me as well, you have to be passionate. And I think you can see the passion from all of the laureates today. And then my final thing that science can be fun. So don't forget to have fun.
I couldn't agree more. Those are two amazing answers. And while you were sharing them too, Dr. DiCarvalho put the usefulness of useless knowledge, which I also love as a phrase. Thank you so much for that. Jeez. Ladies, thank you so much for that beautiful answer. We're going to head to Dr. Briand and Dr. Struhl for our next student question, which hopefully is going to play a little more nicely than the last one. Wish me luck. Take two with feeling. Here we go.
What challenges have you faced in pursuing STEM, and how did you overcome them? What are you most excited about the future of science?
Very nice. All right. Dr. Briand, you're first up. Come on in and what do you think?
Sorry, I did not pick up the question.
Oh, we'll play it again. Sorry about that. Here we go. I'll do it this way.
Have you faced in pursuing STEM, and how did you overcome them? What are you most excited about the future of science?
Nice.
So I still don't understand very well the first part of the question. Yes. The difficulty, or what?
Yeah, what challenges when you were becoming a scientist? Were there any challenges you faced as you started becoming a scientist, or what, or not? If that's true too, that's fine.
No, I had no special challenges in becoming a scientist. Actually, I was always driven by curiosity, and I had medical training. And what maybe pushed me to do research, initially when I started medical school, I did not plan to do research. But what pushed me to do research was that very often we were told, when you see a patient with this and that condition, you should do that and this and that. But I did not get a clear answer. Why should we do that? Why do we do that and not something else? So this led me to look more deeply into some question, and this convinced me that I should start doing research.
Right.
And then during my work, yes, I did face challenge. And I think one of the problems I had is that when you have a really new idea and you want to change practice, you meet resistance. People don't want, physicians don't want to change their practice. They're used to treat patients one way, and if you try to find another way, or initially people are conservative by nature, and I had to face a lot of resistance. But eventually you make it. Yeah.
The classic story in science of this is Semmelweis and trying to convince people of the germ theory of disease. Please wash your hands, doctors, please. And I think it was a very, very long time before people got on board with that. So that story is legion in the scientific field. Beautiful answer. Thank you, Dr. Briand. Gary, come on in. Let's unmute your mic and take us away. Hey.
Okay, well, just briefly, in terms of what challenges did I face when I began to do work in science? And there really weren't any structural challenges. I mean, in other words, it was a welcoming environment for kids and then students who were interested in doing research. I think the challenges are internal. It's your uncertainty about whether you will be able to do it well. And I think what I learned as a graduate student, when I was very unsure about whether I had what it would take to do science, is I realized it didn't matter what I thought about whether or not I'd be able to do it. I should just do it and let the cards fall where they would. And because I couldn't change whether I would have the ability to do it by worrying about it. I should just proceed and let it play out the way it would. And I stopped worrying about that. And that relieved me of a major challenge to my introduction to doing science. So I'll leave it there.
I like the "just do it" featured in the broadcast, our Nike slogan of the day. Yeah, thank you so much for that. We're going to dive in with Daniel with one final question, and then I'm going to bring back in everybody for one final note before we wrap up together. We're going a little bit long because we're all scientific rebels together and in science communication as well. So let me queue up our third question from our student at Visions of Science and get us underway. Here we are.
My question is, how can young people contribute to science before ever becoming researchers?
Ooh, Dr. DiCarello, what do we think?
Yeah, that's a great question. I think as soon as you start to think about a question that you're trying to answer, you're already a researcher, even if you are still a high school student. In a sense, you are already a researcher. So more practical terms is trying to find, some experience in academic labs, some internships in academic labs, so you can start to get your hands wet with the science, doing the experiments and start to do things. I think I would recommend people to start trying to build stuff. Right? You can build a dashboard about, we had the pandemic a few years ago. Can you build a dashboard and start getting information from different places and combine them in a single place that can be useful for everyone else? There's a lot of little things that you can start to do. A lot of data is so much data available today that you can start to try to play around. In the early days, it's very good to try to reproduce. So if you can see a paper and you see that data is available, can you just generate the same figures in that paper by yourself? So you can start to learn how the science process works. And that's a really good way to start contributing. You're already reproducing someone's results, so making it more reliable, but at the same time, you're learning yourself how to do. So there's a lot of things, but my main point is if you are already interested in science, you're already a researcher. You're just in the very beginning stage, but you're already a one.
No better message than that. And I want to note the two programs that we highlight to our students all the time are Zooniverse and iNaturalist. These are tools where you can sift through huge realms of data collected from community members and scientists all over the world and make real contributions to science. So if you're keen on anything from astronomy to conservation to cell biology, there's a lot of cool projects there, and I can't recommend them enough. Fine people, I'm going to bring everybody back in. You are rock stars, and it's been such a pleasure having you all on today. I could go all day with this, and we've already got more questions than we could possibly answer in one broadcast live, but I'll wrap up with all five of you by asking if there's a final message you want to leave our classes with today. Anything before we wrap up and say farewell? Daniel, you're first right beside me, so I will start off with you again. Anything before we say goodbye?
Well, for me, when I was in high school, at some point I was thinking science was all about memorizing facts, and I liked, but I didn't love at that point. Once I realized that science is actually about discovering the facts and making new facts for everyone else to memorize, that's what the hook happened to me. That's when I got loved by science.
Your turns of phrase are fantastic. Making new facts for everyone to memorize is going to be my guiding principle in life, I think, from now on. That's beautiful. Gary, come on in. What's your final message for us?
Okay, well, relevant to my last answer, I will just say one of my favorite quotes from literature, which is from Melville's Moby Dick, which is, I try all things, I achieve what I can. And I think that's what a mindset that you need to have to do well in science.
It certainly is. Captain Ahab, I'm not going to spoil it for anyone. We'll see how that goes. You should all read Moby Dick when you're done. It's one of my favorite books of all time. Iva, come on in and take us away.
I don't think I have all that much to add. I think both Daniel and Gary have said really important things, and I would just say, stay curious.
It's a beautiful message. It's one we hear all the time. And I do know that it is harder as we go through a big group of people. Andre, you're in trouble, man. By the fifth person, it's very hard to come up with a different message. I know, but I'll put you to the test anyway. Jennifer, come on in.
Yeah, I think the only other thing that I would add is really be coachable. So seek out mentors, be open to feedback, and just have that mindset of continuous learning, and that'll take you far.
Very, very nice. And then Andre, no pressure at all, number 5?
No, my advice, actually, it has been given already, but in some way. But talk to colleagues, especially to colleagues from different disciplines. It's incredible what you can learn from them. And also, it has been said already, I mean, discovery often are taken at the convergence of two different disciplines. So talk to colleagues.
No better messages than these. You five are truly amazing. I've had a ton of fun. My face hurts from smiling, which is a great problem for me to have as a broadcaster. I really want to encourage our whole audience, check out Gairdner.org, check out Gairdner Awards. They do celebrate amazing people, as you've all had the chance to see today. This program, like all our others with them, is on our YouTube channel. We've got a special Kahoot series and some great quizzes with them as well that you can check out. And with this unwieldy URL, if you want to join us on the 30th, we've got a second virtual roundtable as well. A few more amazing researchers to feature there. But a big thank you to all of you. We'll wrap up the broadcast there. Go back to saving the world and making it a better place, you five. Have a wonderful day. And bye for now, guys. Thank you so much. So long.